Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Roslin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions to our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Thomas Page from FUJIFILM Diosynth Biotechnologies.
Thanks for the introduction, and thanks for everybody for joining us today. So this is going to be quick moving, so let's jump right in so we can maximize use of our time. So what we really want to focus on today is the importance of flexibility and high containment for these advanced therapies, manufacturing, an application of closed systems with appropriate engineering controls. We won't talk too much about facilities as equipment. That's another concept that's inherent here. And then the main thrust is on the advancement in the fill finish area for these products. So for Fuji Diasynth, the way we try to approach working with our clients, since we're a straight CDMO, is we really want to partner with them, and I like that because it's kind of a pun. And really, the obvious one is about the lives of the patients, and obviously that's of the most important level. But really, being an old CMC person, it's really about the life cycle of the products, and really about advancing to that commercial quality CMC in the most efficient way possible. And so from the very beginning, approaching everything from a risk-based approach, and from early phase, have that continuity into commercial manufacturing. So when we start to think about advanced therapies, there are some really critical differences between this and traditional recombinant proteins, and it can create some little blind spots for us. So a couple of critical tensions that come up as we deal with clients. There's oftentimes a tension between getting proof of concept versus long-term, and that often comes into play when we're talking about being in, let's say, an inherent methodology. When the market, to properly serve the market, you would need the scalability of the suspension approach. I think the other thing that's really critical here is that as the product approaches a cure, that completely changes the profile of the product in the market. And what that really means is you have a large installed base, and for both commercial and for humanitarian reasons, once a product is approved, you need to treat those patients as quickly as possible. So flexibility and scalability is really critical, and the scale is also difficult to manage through to commercial, because commercial products might be in hyperstack or 50 liters, or it might be a fleet of 2,000 liter bioreactors as well. And then the most important take home is that no matter whether you're going to manufacture internally or whether you're going to outsource, these products are going to be made in a multi-product environment where understanding cross-contamination control and the risks inherent with that are really critical. So those need to be designed into your process and your methodology. And of course, the most critical of which is around the drug product piece. So we looked in this market. So we came up with a sort of a proposition where we wanted to go forward in advanced therapies. So we wanted a technology which had legs into the future. That for us is really critical with our risk-based approach, is to absolutely minimize the risk to patient as well as the operators. And then we're sort of looking at batch sizes, typically not more than 10,000, but with a stretch capability to 25,000 or more, and then ability to scale out beyond that if necessary. And we wanted the flexibility for vials, syringes, cartridges, and we assessed the small modular fillers. that fit that profile. And what we ended up purchasing was the Vanarex SA25 Aseptic Workcell. And the reason for that is we used a risk-based advanced equipment procurement tool. We had a multifunctional matrix of very senior folks within our organization, from quality to operations, engineering, EHS. And we deduced with this tool that this is by far and away the lowest risk to patient of any available system. The primary reason is it's closed, but secondarily, it removes the contact hazard pathway for cross-contamination between batches. And it's the only one that we know of that's capable of doing that. And I'm going to show you in a video in a second to illustrate how that is achieved. So it obviously removes the operator from the sterile environment. It also supports the containment requirements, because we can VHP the components in and we can VHP the filled components out. So for instance, you don't have any aerosols on the outside of containers that would then have to be dunked or wiped, etc. So it's designed using risk-based tools, and you have a fully integrated validation approach. These are very high-value products. So on the commercial side, we wanted to come up with an approach that we can work towards near zero defect rates with incredibly high rates from the bulk to the vial. And so when you think about that, you get a really huge net present value from going forward with this, and then obviously flexibility. So let me show you a quick video to show how it works. So the Workcell actually has two isolators. It has what's called the DSI, where you load ready-to-use pre-sterilized components in the tut and nested format. You close the door. Then we perform a 6-log VHP cycle to decontaminate the outside of those pre-sterilized components. And for the aeration, it's brought into the filling part of the isolator. So there are no humans involved, and there are no glove ports on the machine. That red light and flash that you see is the vision system above. So there are cameras strategically located within the roof of the isolator to look for critical aspects of the components. So right there, it was looking for the presence of that slip sheet. On the fill pedestal, it's actually looking for the pattern of the vials or any missed vials. Any of those things that do not pass, it rejects the entire nest. So this is showing you it's capable of doing IPC. So you can see that each vial is being lifted up on a precision balance, tared, and filled. So we can do 100% down to what we typically do, which is 2 to 3 vials per nest. So that's typically in the range of 2 to 5%. So after the vials are filled, they're placed under the closures. So the stopper is pre-integrated into the cap, and you'll notice that the machine is only touching the nest. It is not touching the stoppers anywhere in this process. So it goes into the closure chamber. That's a vacuum chamber, so you can do gas exchange in there. Now that stack is now completely sealed. So that is a fully filled, closed vial system. And now you're reversing out the process. So this is simulating the end of the batch, what you see here. So that filled nest of vials then goes back out into the feed isolator. And for advanced therapies, so live viral agents, we would then perform a VHP cycle to decontaminate the outside of the containers. So critical changes here, there's no crimping. You have a pre-integrated closure system. So it's a little bit like going from an analog to a digital process, and it's amenable to quality by design approach, and we'll come back to that in a second. So the components themselves come in the ready-to-use format. So right now we have tooling for 2, 6, and 10R vials. We can add syringe and larger containers as necessary. We're currently using an closure from a company called A. Ramond, and we're using West stoppers for this as well, and then a variety of stoppers are available that meet the ISO requirements. So the Matrix Alliance is a group that was formed to create standards for these ready-to-use nested components. So an example, SHOT, this will be an example from SHOT, though there are other vendors that do this. It's just to have a nice graphic here, so I like to use it. So you have a standard tub, and then you have 3 different nested type of formats. So you can have the ready-to-use syringes, you've got the ready-to-use vials, and you've got the ready-to-use cartridges, which we can create recipes within the automation of the filling machine to successfully fill. For vials, the press fit closure is designed with a geometric error stack up, so that once this cap is in place and you have this clasp coming below the flange of the vial, you're closed. So you're either unclasped and you're open, or you're clasped and you're closed. I particularly like the A. Ramond style because it actually has a dual layer closure. So you have a cage, and then you have an external collet which rotates on this. It's kind of almost acts like a bearing. And what's really great about that is you can then spin the outside of this, so it is physically impossible to apply any mechanical forces to the seal interface, unlike traditional crimped vials. So that's a really fantastic feature. So we host this in a 3rd generation advanced mobile cleanroom, which creates a viral containment zone, which we run this in a grade C background, and we run it in a negative pressure to the gray space. And then we do the filling within an isolator that does not have any gloves. Now the glove thing is really critical because if there are any issues with filling, you know about them at the time of filling. You're not finding out after that you have put all of your product at risk to find out that there's a pinhole in a glove, which is obviously a terrible experience, and most all of us have experienced that, previously in our life. So importantly for this technology, it's really critical, and this came up in our risk assessment, since we're extensively using vaporized hydrogen peroxide to perform decontamination cycles, both going in and out. We had to guarantee that we would not have any residual peroxide in components. So we adapted an impermeable lid to put onto the components, and this has an aluminum foil inner layer, which we have validated to prevent peroxide ingress into the components. And we did that using a very sensitive virus. So we did spiking studies to look at the response curve of peroxide on the virus, and then we used that live agent to do studies within the filling machine. So we remove that possible hazard pathway to the product. So what's also great about this, there's numerous companies that have brought in this technology, and Vanarex, along with the Matrix Alliance partners, have created industry working groups. So we are all working together collaboratively to push this design approach forward, because it has the potential to radically decrease risk to patient within this technology. And we know that it will happen much like what happened in the drug substance world with adoption of ready-to-use components and process closure. And we're working really diligently to achieve this near zero defect rate. So in the conclusions and challenges section here, we're basically say that we're delivering the lowest risk that we can to the patient and the product and to the operators while maintaining a very high value proposition. And it is often the case that new technology would enter into high value use and high risk use, and where the status quo does not adequately control those residual risks. So we are working along with these users to continue to drive out risk, mostly at this point through advanced specifications around components. We are working very diligently to achieve what I call closure by design. So that way you have guaranteed closure once the cap is in place, no matter what the conditions. So, and it's really critical to set that specification so that the components are interoperable across vendors. So we would want OMPI, SHOT, SiO2 MED to function. We would like Datweiler and West stoppers, and we would like multiple closure designs, so that it is not incumbent upon the user community to create those validation activities, so that it can just be widely adopted by the industry. We are designing that to worst case user requirements for ocular and pediatric, for example. And then the interoperability and standardization is critical. And then the last piece we're working on, which we should be vetting out this week, is to get our batch loss volumes under 10 mLs of loss. And currently we're at about 2.2% reject rate. We've identified the cause of that, and the solution to that should be in place so that in the first quarter next year, we should be achieving our target of basically zero defect rates. And then the last piece is we're working on high throughput versions of this as well. So I hope that was somewhat informative, and I think we could move to the questions.
Okay, great, thanks. So the first question is, what is the most important outgrowth of this approach? Oh, well,
I think that's a good question. So basically, for me, the point is that this is an outgrowth of risk-based design and risk-based operation. And I think the most important outgrowth of this is standardized components that massively reduce risk to patient in the order of million folds. And I think that is really critical, because there haven't been any sort of great leaps forward on the drug product side relative to the drug substance technology. So I'm super excited about this.
Okay, great. So can the system accommodate bags as the primary DP container?
So I asked that question recently to the vendor. There is currently, it is not available, but they do have a way to make that happen. We just haven't had a driver to go down that road, but I have been told that it is possible to do, and that there is an approach to do it.
Okay. And we have a couple questions about product changeover. So one is, does the isolator need to be decontaminated? And the other is, what cross-contamination risk mitigation steps are taken for the heads?
Yeah, so basically the product contact tubing is all disposable, and the tubing is a coaxial design that prevents VHP ingress, and then the fill needle comes sheathed in a holder, which is sealed. The machine itself will not let you run unless you perform a validated decon cycle in the main chamber as well. So as you would expect with a robotic system, it's characterized by numerous interlocks in the software to prevent you from operating outside of the qualified approach. And then there's multiple user login levels, so the operators actually can't get around any of those interlocks. So one of the reasons we picked this is because it has the least risk of cross-contamination that's out there. Because there's no contact hazard pathway, that means like in a traditional line, you would be dropping stoppers into a stopper bowl. So if you look at the Annex 1, even if you're in an isolator, VHP is not sufficient. You have to autoclave that bowl and then figure out how to introduce it into the chamber successfully. That is not the case for this technology because there's not even any indirect contact with the sterile component. So that means that it's really really great for live agents in particular, but great going forward for other types of products. And we are looking to extend this offering. We're buying another machine to put in place for antibody fill finish as well in a different part of our facility, a different facility actually. And so yeah, you inherent to the operation of the system, you're performing numerous VHP cycles to decontaminate. And then we have developed a cleaning approach to remove particulate, and we actually use these crime scene rollers that the FBI uses to recover fibers. So that helps us remove any particulate from the environment, and it acts as a cleaning approach and also as well as a detection method. So it does double duty.
Okay. Well, just in the interest of time, I think we have time for just one more question. You can go ahead and continue to type questions, and if we don't get to them right now, they'll be passed along to Thomas, and he can follow up with you directly. So the last question is, what throughput rates have been achieved, and how long have product changeovers taken?
Okay, so right now for a 2R filling, we can basically operate in the range of about 1,000 per hour. The product changeover is very fast. You can change out the tooling in an hour, which is quite nice. We did some development activities with this across different components, and we basically did 5 different recipes in the same day, which is more or less unheard of. And what's nice about that is you can operate this machine with 2 people, and there's no aseptic gowning qualification, and there's a very fast changeover capability. So I didn't hone in onto that bit, but the flexibility angle of this is really incredible. This week we did a series of engineering runs with the product where we were screening different component sizes, different stoppers, different materials of construction, different fill targets, and we did 2 engineering fills a day with our staff. So very versatile.
Okay, great. So thank you, Thomas.
Thank you, and I appreciate everyone for tuning in today. A special shout out to Guy. I see you there. So hi, Guy. So thank you, guys.
Great. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We invite you to join us for future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox.